we'll wait about 1 more minute and then we'll start. In the meantime, please feel free to introduce yourselves via chat. We'll
Okay,
also use the chat to ask questions, or you can also raise your hand at the end
I
of the.
want to introduce myself. I am Oluwafemi John Akintayo from Datican Project, Lagos State
Oh,
University in Nigeria.
can you hear me? Hello?
Yes, I can hear you. I can hear you clearly.
Oh, I
Can
was
you
going
hear
to
me?
say that we don't have enough time for everyone to introduce themselves in voice. That's why I said chat. Because there are too many of us for us to go one by one. But it's good to meet you, Dr. Femi. Okay, so I believe we can start now. I would like to introduce myself. I'm Dr. Nafisat Oki. I'm a program director at National Institute of Dental and Craniofacial Research at NIH. I manage data science, computational biology, and bioinformatics program, and I'll be program portfolio, and I'll be your moderator for today. We have two presentations in this session, one from DSI Africa TB, and the second from DSI Malaria. And I believe both Dr. Katiti and Nakasi are here, so we can start with the presentations. So is Wisdom here? Will you be taking care of showing the recordings? Okay.
Hello everyone.
So this is the
My
first
name
one.
is David Katete from Makerere University. I'm going to discuss about DSI Africa TB project, acronym that way. I'm one of the MPIs. The study team represented some of the members, say multidisciplinary team of investigators, computer scientists, health data scientists. We are all from Uganda. And the rationale behind this project is the fact that there have been questions that are lingering in TB despite it being a known disease, one of which is a complete understanding of the mechanisms allowing complete eradication of M. tuberculosis, the etiological agent, in a few individuals once infected, and of the mechanisms failing during latency, thus leading to reactivation of disease in only a fraction of latently infected people. We also have poorly characterized disease phenotypes. It's no longer the usual two dichotomies we used to know, latent vis-a-vis active TB. We also now have subclinical or silent TB. And in Uganda and Africa at large, we have people who can resist infection despite persistent exposures. The characteristics of individuals underlying all those phenomena is not known, nor the biological or immunological questions. They remain unknown, and that's why we aim to leverage machine learning approaches and build models from the existing databases to attempt to understand, attempt to resolve some of those questions. There are two aims. The first aim is to predict patients at baseline who won't bacteriologically convert at months 2 or 5, hence at risk of failing treatment. Those time periods are key in TB because when a patient does not convert, we can know that they are at risk of failing treatment. And M2, we are trying to identify the characteristics of contacts who could resist TB, or those who are at risk of developing TB disease. And these are contacts who stay with the TB cases, those whom we call the index cases. The approach is summarized here before. We consider an household, a community with cases and contacts as envisioned in this figure. And then we got data sets. We have TB cohorts, which have been collecting data sets, clinical, demographic, laboratory data sets, the data sets that are leveraging to identify patients who are characteristics of patients who can resist, who can fail treatment, or even the characteristics of contacts who could resist infection or who could succumb to disease. We are using available data sets from available cohorts, and then we use those data sets to build models, machine learning models, and attempt to identify those patients and attempt to answer the aims. So, but we are also interested in more data sets. We are analyzing and attempting to include some more data sets, even program data in Uganda, for example, the National Tuberculosis Laboratory, the Kampala City Council clinics, and the Mulago Referral Hospital TB clinics. Mostly these are drug-resistant monitoring data sets. We are also attempting to include those into our models. So we have a teamwork that we ensure that we all work together to ensure the success of the project, from the secretariat, the clinic, the laboratory, and our computer scientists. We are all well coordinated, and this ensures success of our project. And one of the pathways to impact, once we answer our aims, we have a technology partner in Uganda called Emergent AI, whom will give our data, our answers, and then they attempt to build the applications that will be given to another project that deploys new diagnostics in Uganda called FEND, Feasibility of Novel Diagnostics for TB in Endemic Countries. That cohort tests new TB diagnostics, and we will work with them to provide our applications that will have been built by Emergent AI to deploy on the TB patients in an attempt to see how they work in the real world setting. And in terms of activities, those in black are the ones that we have either finished or working on. And we, by the end of the second year, we would have answered the aims and also touch on the remaining activities in orange. And I would like to acknowledge, thank the Coordination Center and our funders and NIH.
Thank you, Dr. Katiti. We'll hold questions till after both presentations are done. And you can start asking them in the chat, by the way, but we'll hold them until after both presentations are done. So up next, we have Dr. Nakasi. Thank you, Dr. Nakasi. Please start the recording. Wisdom, can we start the recording for Dr. Nakasi?
I think Dr. Nakasi was supposed to. Okay, let me just.
In the meantime, if anyone has questions for Dr. Katiti, please go ahead and ask now, since we have time. Is David here? I have a question. David, are you here? I know he was here when
Yes, I am.
Okay, all right. So As you were, I guess my question is more around your methods and participants. Can you tell us more about that process?
Oh, thank you for the question, Dr. Oluwafemi. So the participants, they are mainly in two categories. We have cohorts, research cohorts that are going on in Ugandan referral hospitals and at Makerere universities. These are TB cohorts that have been here for a long period of time, more or less decades, and they have enrolled TB patients, and they collect various types of data sets from clinical demographics, laboratory data, can be microbiological, or data related to the host, imaging, chest X-ray images, all that kind of stuff. So the data sets are they are collected from those cohorts by us. We request the data sets, and those are the ones that we build machine learning models from. That's kind of one aspect of the patients from those cohorts. The main one, actually. The other aspect of the data sets we aim to get from program data. These are patients that are looked after by the ministries of health, and these are mostly drug-resistant TB cases. So those are the patients and from whom we are getting the data sets. And the method, so they're about building machine learning models, and most of these cohorts or the programs, once the patients are enrolled, they are followed, and at each of the time periods, there are additional data sets that are collected. And we also get those follow-up data sets from the follow-up periods, and we add them to the models. I think essentially, I hope I've been clear, but I can clarify again.
Just a follow-up to that question. You intend to go out to the community and do intensive case finding, so that where you pitch up tents in the community and see those who are not able to reach these two areas where you fetch your participant from.
Okay, thank you, James. Actually, we don't go to community, but these cohorts, the model they use, they are using a household. They are cohorts which define a case, a TB case, and these are cases in a household, of course, household and the communities of Kampala. And then in that household, say that index case lives with about 5 or 6 people. That household, we go to the household and screen, or the cohort would have screened for the contacts, and the contacts and triage them, either they have TB or no TB. So we look in the records. For the healthy contacts, we recruit those who were screened as not having TB at baseline, and we collect data sets as they followed them all through, and we try to analyze if they succumbed for TB moving forward or not. The same applies to the treatment response monitoring. So the model we use is a household model, but we don't do active case finding. Thank you.
Oh, okay. Any more questions? I have one more, and then we'll move on to the next presentation, and then we'll continue with questions after that presentation is done. But my question is, how do you measure or assess reinfections? How do you, for each participant, how do you track if they do get more exposures post the first time you meet them?
Good question, co-host. Actually, we rely on what the program does. If it's the program case, or if it's a cohort, we rely on their definitions, how they do that. For most of the research cohorts, in fact, some of them also, they rely on what the programs in Uganda, the TB treatment programs do. In treatment response monitoring, they get additional samples once a patient is treated. At month 2, they get sputum from them, and they could do gene expert on them, or they could do microscopy and they try to see whether the bacilli are still there. Even month 5, they do that. Gene expert or microscopy to see if the bacilli are still there. If the bacilli are not there at any of those months, then they declare the patient converted. So those are cured. If they are there, then they haven't converted. So those patients are those are the cardinal patients for treatment failure. That's how we do it. But for the contacts, they are routinely screened by blood. These contacts were healthy at the baseline. At those months, they take blood from them and they screen them using the interferon gamma release assays to see if they have signs of infection for tuberculosis. It's not the best assay, but that's the best one we can use for now. That's what is done at the moment.
Okay. Thank you very much. I think the second presentation is now ready to go, and we'll go to that, and then we'll take more questions afterwards. Thank you all very much.
Hello everyone, I'm Dr. Rose Nakasi from Makerere University, the principal investigator for the DSI malaria project on automated mobile microscopy of malaria diagnosis and surveillance in Uganda. Behind this project is a team from Makerere University, from Sunbird AI, from Boston University, as well as the different health facilities that we are collaborating with. The motivation for this project stems from the disease burden. Malaria is one of the life-threatening diseases, and this is caused by a parasite that is transmitted to people through the bites of infected female Anopheles mosquito. It's one of the top 10 causes of death in the sub-Saharan Africa, and globally it accounts to over 3.4 billion cases. Specifically in Uganda, malaria has been reported to account for over 27% of the deaths, and this signifies the burden of the disease, especially in the sub-Saharan Africa. But one of the key challenges that is faced in the highly endemic countries is one, the inadequacy for microscopy diagnosis for suspected patients, but also this is largely due to lack of enough skilled technicians to operate the gold standard mechanism, which is by the use of the microscope. From our project rationale, there are different existing automated systems that have been developed elsewhere, but the challenge that these also pose around the solution, especially in the highly endemic settings, is that one, they are limited in impact, as well as especially in the highly endemic and resource constrained settings, where the cost and ease of use are one of the major drivers for any solution that can be suitable for a context such as that. Furthermore, when you look into this different surveillance approaches in East Africa, for example, to be able to track and monitor diseases, for example, in Uganda we have the DHIS2. These are constrained because one, they report weeks or months after the disease epidemic has advanced to probably worse stages. Through the DSI malaria project, we aim to produce a functioning patient-level point-of-care diagnosis system that is able to work on the principle that one, we're looking at a solution that is going to be cost-effective, that is going to provide ease of use, but also tailored to a setting such as that that is resource-constrained, and it is also capable of running on multiple microscopes and different phone combinations that are available on market. We also want to further combine that mobile-enabled automated diagnostic system with the spatial and temporal capabilities, and this is for real-time surveillance of the disease. So through the project objectives, we aim at developing machine learning approaches for the standardized field-based microscopy of malaria in the country, as well as building a complementary framework for real-time disease surveillance and improved diagnosis of malaria platform through the different in-field diagnostic pilot studies that we are going to be conducting. So for the strategy for the automated malaria microscopy comes from the fact that one, we want to capture every field setting to be able to look at the specific conditions and settings, and these may include things like what are the different smartphones that are available with their different resolutions, the different microscopes, the different staining materials and stage micrometer readings, and we want to calibrate all this information and be able to infuse it and calibrate it to the different machine learning models that we're going to provide. And the essence here is to be able to come up with a solution that is going to be able to fit the different microscopy settings across the country. So the outcome here is that we want to produce an ease of use AI-based malaria diagnosis tool that is deployable in the different representative clinical settings in the country. So our proposition here is to be able to, one, we have developed a 3D printable adapter that is providing an attachment of the smartphone onto the eyepiece of the microscope in sync to be able to capture whatever is under the microscope and bring it onto the smartphone. We're curating as many images as possible, and then we apply machine learning models to be able to capture the different objects of interest. For this, we are looking into all the different aspects of malaria microscopy, which includes parasite detection, parasitemia determination, different life stage identification, as well as species phenotyping and identification. For our strategy for malaria surveillance is that we want to be able to develop models that are based on risk map information priors for improved malaria diagnosis. And the idea is, once we have been able to detect, for example, a case at a given healthy facility, can we be able to seamlessly report that into an electronic health management system to be able to come up with real-time risk maps, and then we can aggregate that information with any kind of information that has an effect to the spread or to the prevalence of malaria. So we want to be able to look at different pieces of information. Climate and environmental conditions have a very high impact on malaria prevalence. We want to be able to capture all that through the location information, through the social demographics data, through the malaria intervention records, so that we can be able to aggregate all that information to come up with a holistic malaria surveillance system that is timely. And the outcome of this is that we want to be able to have malaria real-time risk maps generated, as well as the different spatio-temporal malaria models. The whole idea is to come up with timely information that can be relied on by the relevant stakeholders to direct the necessary interventions across the country for the different patients. What we've been able to accomplish as a project is that one, we've conducted a fact-finding survey in over 30 select health units in 4 main regions in the country, that is Eastern, Western, Northern, and Southern. And the idea was to be able to understand the ecosystem of malaria microscopy, how it is conducted, and this also guided on the selection of the different healthy facilities that we are currently collaborating with. We've also further held a project launch as well as a stakeholder engagement workshop, and the idea was to be able to enlighten the different stakeholders about the project objectives, the project impact, but as well as what we've been able to accomplish in the past to appreciate the solution, especially around the entire public health domain, specifically around malaria diagnosis. We've curated one of the most comprehensive malaria data set that is publicly available, and as well as in-house at Makerere, that has been collected over the past decade. And across this, we've also developed machine learning models trained on those curated publicly available data sets to understand what has been done in the past around data and around models, to lay ground for any improvements that we want to come up with in the current operations. And that has actually given us better insights on what data to collect and which models that we shall rely on for the new data set that we're collecting. To do that, we've also been able now to train microscopists, especially for those new healthy facilities that we've onboarded across the country, to enlighten them about the data collection tools, data annotation tools, but as well as the project in general. We've collected and annotated data from the 20 healthy facilities currently across the country around thick blood smear imagery and thin blood smear imagery. We also holding stakeholder engagements in the different healthy units that we go to, first of all to make them appreciate, but also have a ground of them appreciating the solutions that we are trying to come up with. The project needs currently is that we're looking into more data, and especially for the surveillance module, because we want to be able to integrate data such as the case and mortality data, climate and environmental data, socio-demographic data, malaria interventions, to infuse all those in the disease surveillance models to come up with more realistic data. It may not necessarily be from Uganda, but across the sub-Saharan Africa, that may help us gain insights and build robust prevalence prediction models. We're also looking into aspects of computational resources. Currently, we are relying on the cloud credits, which are not forever, they are timely. So in due course, we may need a permanent solution that can come in forms of GPUs, servers, and all that, but as well as more cloud credits to be able to support the computational needs for the project. We also want more equipment if it's available, so partnerships can come in those forms where we, for example, right now we are dealing with that 40 healthy centers, and so we need microscopes, we need phones to be able to reliably undertake this project. Below here is a project, different project activities, those that we've finished and those that we are about to finish across the 3-year project. But most importantly is that at the end of the day, we want working solutions that are tailored to the different field settings. I want to acknowledge the DSI Africa Consortium for giving us a platform such as this to be able to air out our research, but also the support in undertaking this research has been very pivotal and timely. I also want to acknowledge the support that's coming from our funder NIH for supporting to be able to undertake this study. I also wish to acknowledge our partners from Sandbird, from Boston University, that have been pivotal in running this project. Thank you very much for your attention. You can reach me out, but as well as on our different social media handlers. Thank you.
Thank you very much, Dr. Nakasi. And I guess virtual applause for both you and Dr. Katisi, sorry. So, and now we can proceed with more questions, if anyone has any. And please remember, part of what we're supposed to be doing here is not just talking to the presenters, but also talking to each other. So don't forget that this is also a networking opportunity for everyone here. But if you have any questions for Dr. Nakasi, she'll be able to take questions now. She's also here in person. So I guess I can start with one question for Rose. So the methods in which you're using for your analysis, how would you compare them to, say, a gold standard method? So what you're using now for the blood analysis, is there a gold standard method? I guess I should ask. And then the other thing I guess I'm interested in is, given the spread of the participants and what is the fault of the participants and the time and how long it takes for the disease to go from incubation to actually being manifested, do you feel that this surveillance tool can catch people at the key times for which interventions can be made? And what sort of interventions do you think could be made? I think I asked multiple questions, but feel free to answer the ones that you're able to.
Thank you very much for those wonderful questions. What's the gold standard? Well, rely on what WHO guidelines put forward, and currently what's stated as the gold standard or the confirmatory test for malaria diagnosis is by the use of the microscope. How the current interventions through artificial intelligence in terms of the gold standard, I would want to rate it in form of machine learning or artificial intelligence being a support system to the gold standard, because currently the gold standard is microscopy, but it comes with some shortcomings. For example, the timelines it takes a given technician to be able to view and analyze the different pathogens under the microscope could essentially be longer, but also coming from the fact that we have very few trained technicians to even man that gold standard is another limitation that comes with the gold standard itself. So in how we would wish and how artificial intelligence is actually supporting is that especially when it comes to the examination or this analysis of what is under the microscope, the artificial intelligence is able to do this with a lot of ease, again, in terms of consistent accuracy that is non-subjective to a given technician. Also, that comes in very handy. We're also looking at a solution that is going to be cost-effective in the long run, because where a technician or a pathologist has been screening or allowed only to screen less than 30 patients in a day, the artificial intelligence is able to go beyond that. And that means that we're going to have a bigger turnaround in terms of the number of patients that are going to be reviewed in a given day. And the ultimate goal is that can people get timely diagnosis so that they can get the relevant medication to go around issues of drug resistance that are very common, especially around malaria treatment currently. So that is those are some of the major things that we look at where artificial intelligence comes in handy. It's not so different from the gold standard, but the idea is it is actually a decision support to the current gold standard mechanism. I don't know if I caught other questions, probably just re-echo one or two so that I can be able to respond to those. Thank you.
Sure, thank you very much. I think the other question I had, I would just ask again later, but because I see some questions in the chat, so I want to get to those. There is one from Oluwaseyi Ashaka, and they said, thank you for your presentation. I have been looking forward to using this approach for surveillance and for modeling the same impact in AI-assisted pap smear microscopy. Is this model scalable to other microscopic diagnosis?
Yes, obviously, and actually we have scaled it up to tuberculosis. We have scaled it up to cancer, cervical cancer, and specifically around actually pap smears, and as well as the histological detection of cervical cancer. That's looking at now the biopsy smears. So we are trying to build a benchmarking solution that is going to be scaled across all microscopy disease challenges. Thank you.
Thank you. And I see Oluwafemi Akintayo hand raised.
Okay, thank you very much, Ma. I want to ask questions concerning the malaria diagnosis. So concerning the types of model which you wanted to build, which I saw. So I saw some on prediction of the outbreaks, prediction of the contacting of malaria. Now, could you please give us some kind of insight about the data sets that you want to use? Are they clinical data sets or imaging data sets or combination of both? I'm struggling to understand that aspect very well. Could you please shed more light, ma'am? Thank you, ma'am.
Thank you for that. So I don't know whether this is specifically for all you trying to combine the two for both diagnostics and surveillance. And one of the things that we're trying to do is actually somehow both. And one of the proposition is that can we be able to use one surveillance to augment our diagnostic model? And in this case, we're relying on, for example, spatial data. Even CNNs can be able to do that, to integrate different data sets and then build robust models that if, say, for example, a person has been diagnosed of malaria through our diagnostic model, can we be able to rely on any other information from the environment to be able to also augment? So convolutional neural networks have that capability to be able to combine data from different sources to give more insights into what the diagnostic model has done. But also in turn, it is actually also diagnostic results are also in turn informing the spatial temporal models or the surveillance models, in a way that if someone has been diagnosed, then the case is captured and risk mapped and tagged to a given location. And then there we can go into the different modalities where we again combining different data sets from environment, from social media data, and all aspects that may have an effect towards malaria surveillance. Also case data from the DHIS too. So that is also going to be one of the things that we shall rely on for the surveillance model. But this will only be used for us to get counts in a given location, or at a given healthy unit, to be able to understand the prevalence of the disease within a given setting. So there are different machine learning models that can be able to combine that to be able to do that. But CNNs are also very powerful in doing that, away from just the traditional machine learning like the random forests.
Thank you very much, Ma. I really appreciate the concept of the convolutional neural networks, which you have just mentioned. So could you please enlighten me more? Because I saw two types of images, the microscopic images and also a picture of the mosquito bitten face that was actually annotated like that. Could you, have you been gathering any form of data set, either the microscopic or the physically beaten individuals? Please, I want to know that, ma. Thank you.
I think I need to put clarity on this. Specifically for the diagnostics, we are relying on microscopic data. The image where the mosquito was is just to put in context of how malaria actually gets spread. We have another module altogether where we actually also looking at mosquito identification, but that's away from the scope of this project.
Okay, you have answered my questions, ma. Thank you very much, ma.
Thank you, Rose. I actually do have one more question of my own. What interventions do you think could potentially be applied based on the findings? What actions do you think stakeholders or governments, health agencies can take based on these outcomes?
Based on this outcome, and I'll communicate this, especially coming from how microscopy is conducted, and again coming from the experience of the survey that we carried out in the different health units across the country. They gave us an idea of actually how microscopy is being conducted, especially at the lower healthy centers. You will find that mostly what they are using are mainly RDTs. So we wanted to identify very quickly the patient information, patients that are positively having malaria. And this was done through one, getting information from the RDTs, but exposing the same to microscopy, what we identified and found out was that out of, for example, 20 patients that have been exposed to RDTs, you'd find that the disease, the diagnosis when it's exposed to the gold standard, which is by the use of the microscope, is either a positive for a negative and a negative for a positive. And out of the 20 patients, you'll find around 10 actually giving false negatives or false positives on the microscopy. What that then means is, if that is the kind of diagnosis that is being utilized, especially at the lower health facilities, is that we have a very bigger burden that is going to come from the diagnostics. And therefore, what is mainly stopping the gold standard being used at some of these healthy facilities is, one, you would not find a WHO certified microscopist who is on ground to be able to man the gold standard. Then what that means is that so many patients are going to go with poor or wrong diagnostics, and that means even when it gets to medication, they are going to either receive the wrong medication because they'll be treating something that they don't have, or they'll be getting the wrong dosage for a given different other disease altogether. So coming from that experience, and from the work that we are currently doing, I do believe that it's one step at a time, but I know it's heading in the right direction, because if we are able to improve the gold standard currently, then and increasing also the throughput or the number of patients that are going to be diagnosed accurately to the system, I believe that we are going to have better patient outcomes at the end of the day with the right medication. But also, most importantly, is that we don't want to have the disease detected when it has gone to its advanced stages. Can we have a diagnostic solution that is embedding aspects of accuracy, timely, but as well as cost effectiveness? And I believe that this is one solution that could go a long way in supporting some of the limitations that we have, especially in our context. Thank you.
Thank you very much, Dr. Nakasi. And if you have more questions, please feel free, raise your hand, put a question in the comments or in the chat box. I see one comment for Dr. Katiti. David, are you here? I see him here, so I'm assuming he's here. What is your validation and dissemination plan apart from writing of manuscript? This is from James Orwa. And James, feel free to expand on this if you want to.
Yeah, thanks. I think, oh, so you go first.
Pardon, can I go ahead and answer, or James?
Yes, please, David, go ahead.
Oh, thank you, James, for the question. Actually, I didn't clarify that well, but validation is part of the initial process that we have been doing in answering the aims. We have the AI lab, which does the building of the machine learning models. They initially took data sets from around, I think, 100 patients for training and validation, and that process is getting completed this year. And then depending on the feedback, and they are going to scale that up to the entire sample size that we set up in the project, about 600 participants. So we are using data sets from 100 participants, that's patients and cases, to do the validation. But we are doing it. And then after this, we also have a plan to disseminate to the Ministry of Health. Some of them are part of the team, but we have a plan to disseminate the findings, particularly the Central Public Health Laboratories, acronymed CPHL, which is part of the Ministry of Health in Uganda, is so keen in applying these modern methods to surveillance. So we are working with them, but we will disseminate to them and then the wider Ministry of Health at large. But thank you for the question.
All right. Davis Balwire, I hope I'm pronouncing that correctly.
Yeah, you got it right. So I have a
Okay.
question for the two presenters. Is the data available for us maybe who want to practice and improve our skill sets regards machine learning? And
Yeah, if I could go first, the data sets for the TB projects will be available immediately after the building of the models and the dissemination and then the publications. And some of them are even available for now, particularly the sequencing data that have been anonymized. Those are available. Upon request, some are already available as sequence read archives, but the participants' data that's linked to them is not available to those. In other words, data sets, yes, are available upon request, and they will be fully open shortly after completing the project.
Thank you very much. From our end, we still are undertaking a process of data collection, but we intend to actually make that data open source once all the different data collection and annotation processes are finished. And I know that we shall be collaborating with different other individuals who might wish to use the data set.
Maybe if I can add to that, we are also participating in the NIH program called the TB Portal, whereby we are also uploading data sets for each participant. This could be the social demographics or microbiological data, laboratory data. And at the end of the year, the data sets will also be available in the TB Portal as well.
Okay. Thank you very much to our presenters. Davis, do you have another question or is just the hand raised from before?
yeah, one more question. This is regards, is it possible for some of us to volunteer? I mean, the reason for such platforms is like for today is for us to kind of like network with people, meet people we can work with. So is it possible for, let's say, if I contacted them to be able to maybe take part in the projects that they are conducting?
Very much so. And I don't know how on my side how I can enable that, but I'm going to type my email here if you do not have it so that we can communicate. Thank you.
And also from me as well, yes, and this is the reason for such engagements, to be able to collaborate and partner just beyond the presentations here. I had highlighted my emails, but I can also re-type them here as well for any further engagements of the call. Thank you.
Okay, thank you very much to Dr. Nakasi and Katiti. I believe we have now come to the end of our session. And I want to thank you all for attending and engaging in the sessions, and to our speakers. I think at this point, we can now start heading back to the main room. If you have any more questions, please feel free to put them in the chat, and both Dr. Katiti and Nakasi have put their email in the chat as well, so you can reach out to them via email if you want to continue the conversation beyond this exchange meeting. Thank you all very much for your attention and your attendance, and we'll see you in the main room. Okay, bye.